describe the structure, functions and disorders of immunoglobulins with a brief description of cellular and humoral immunity. This is your syllabus, and this is basically the nano form of immunology. This basically covers the immunology as such, you can say. Now, briefly, how briefly I will tell that I really do not know, because you see there is one institute, National Institute of Immunology, fully dedicated for immunology research. This was established sometime in 1981 and this became fully functional at its site at New Delhi from 1986. And I visited this place several times during my MD tenure. So, this is basically a research institute. In addition, you can imagine that some super specialty medical teaching institutions offer 3 years DM course after MD on immunology. And many medical colleges having an immunology department, many books, journals are there on immunology. Now, how briefly I will tell, how many classes will require to cover immunology? You just tell me. You suggest, how many classes should I take? How many? What briefly, your syllabus was brief, describe briefly, description. How briefly I will say? How much? How many classes? Huh? 3, yes. Quite logical, 3. So, 3 classes I will not take, I will take only 2 classes. One will be discussing, basically if we want to know the immunoglobulin, then if I start here, you will not understand this, without understanding this part, this immunoglobulin part will not be understood properly. So, all these things are not required to be learned at this stage or understood properly, but you can definitely, but if whatever I will tell, if you really listen very carefully, at least your 4th year up to 4th year it will be covered and you will understand very easily about the immunology part in microbiology. And these basic fundamental concepts of immunology are there what in my lecture. And in a short while you will see there is a full length question paper on class 10 marks in the second prof examination last year. So, this is the class which I am taking today. So, I will first cover cellular and humoral immunity as briefly as possible, covering many points which are not available in your book. You may not understand everything, but try to note down whatever you can, and I will ask specifically to note down those things you must write down. Rest you understand. And if you cannot really understand, in the there will be SG, I can take one SGT class, and there after reading the books and going through this video or lecture, you can discuss at length during that 2-hour period, 2 or 3 hours, even I can take longer than that. Okay, so I basically my purpose is to orient you or to prime you to understand immunology so that you learn immunology better during microbiology class and of course in clinical practice. So, what I have decided that I will cover antigen, innate and adaptive immune response. There is some new terminology which you will be knowing. If you do not want to listen, I mean note down, you can sit down there because it will be difficult to write down. You have to be very fast. Innate and adaptive immune response, cellular and humoral component of immune system which is there in your syllabus, but without knowing that thing you will not understand that part. And the central role of T helper cells in immune reactions, self and non-self recognition, that is very important in immunology because we can't mount an immunological response against our own body, but that does happen during several diseases. These are called autoimmune diseases when the body develops immunity against its own antigen. And in the next class, I will concentrate on structure, types, function and disorders of immunoglobulin. So, what is immunity? We talk very loosely. he doesn't have immunity, or there are several immunity booster drugs, you take Chyawanprash, you take this thing, that thing, your immunity will be increased. But how do you measure, quantify immunity, whether my immunity has been increased or not? As such, there is no good parameter except counting the cells. There is no observable reactions by which you can say that I have developed immunity against that. Only cell count, only antibody, these are the things which you can observe. So basically immunity is you see the state of being insusceptible or resistant to a noxious agent, usually a pathogen, bacteria, virus, microbe, anything. It can occur naturally because we are exposed to the nature to various pathogens. So it can occur naturally, and as a medical person will be exposed to infections in the wards, in the OPDs, from the patients who are suffering from some kind of infection. So you will get a prior exposure, or by immunization. All of you have been immunized. That means you have been artificially given that particular infective agents in a form by which you developed, or from which you developed immunity. So there are three salient features of immunology, that is recognition of self from non-self or foreign. Something foreign has entered into our system, our body must be able to, recognize that non-self or that foreign element to be dangerous. All substances which enter into our body are not dangerous. So they don't mount any, our body doesn't mount any response against those, but those which are going to cause some amount of harm, for which this immunity is developed. Second is specificity of the reactions. That antibody or cellular immunity, whatever we develop, that must react very specifically. And third is memory. See, if we encounter a good person or a bad person, we memorize that person. Now, next time when we deal with that person, we deal accordingly. So, therefore, that memory helps. Suppose if I know that that is a bad person, well, I will be very much well prepared. Or if he is a good person, then I will be prepared. Similarly, we are dealing with infective agents. Now, I got an infection, I developed an immunity. Next time if I get the infection, my body or every organism's body will encounter that infection very effectively. That is what is memory of the response. That you will, all these things you will, I'll discuss in detail. So there are three levels of difference, don't get confused. We are having a physical barrier. Physical barrier means outer skin and the mucous membrane. You see everything is outside. You look at this thing that is in continuous, that our body, we are having an external skin, and if you go a little close to the nose, nostril, you will see that this is continuous. So this is continuous up to alveoli. Similarly, if you come to the mouth, you will see the oral cavity is there, still it is continuous with the anus through intestinal tract. So basically, that area is basically exterior, that has been invaginated or involuted. So that is a physical barrier. Suppose if the skin is damaged, obviously any pathogen can enter so very easily. Suppose just like a boundary wall of anything. That boundary wall protects someone to enter into the campus. It is like that. So actual immunity is not conferred by this barrier. Don't get confused. It is done by innate immune system and adaptive immune system. And they work in tandem. There are two interdependent entities that you will understand, but they work in tandem. One example I am telling you, our institute is a separate institute altogether, but it is interactive with the West Bengal Health University. in tandem, isn't it? That West Bengal Health University is working in tandem with NMC. So they are all separate systems, but this is just for example understanding. So they are working in tandem, that you will realize also. I will explain all these things. So this is, I decided to tell this thing at the end. Now you note down all these points. This difference between the innate and adaptive system, because I thought that initially I will explain everything and that at the end I will tell you the difference. Now you note down this thing, you just be very fast. Each and every point you will understand during my lecture. So first is time, innate immunity. It is present from the birth, remember. And it is controlled by the germline. What is germline? I will explain. And it is inherited. Adaptive is after birth. It is not at the time of birth. From the mother's womb, we do not get it. That is the reason adaptive is called also acquired. Hence, we acquire after birth. Innate means we are born with that particular immunity from the mother and from our body while we are in the womb. That is innate immunity. So, innate gives a first line defense, but that is less potent. Yes, here you see adaptive is on exposure. We need to get exposed. It is not inherited except placental transfer. And you just note down all these points which I have written. Somatic selection. What is somatic selection? That I will explain. Primary and secondary responses are there in adaptive immunity, and there are immunoglobulin genes, and there is diversity of the immunoglobulin genes that I will explain during immunology class, immunoglobulin class. It confers second line, but it is more potent. Specificity, innate is non-specific. Adaptive is specific. Response, innate, immediate but short lasting. Adaptive, slow, delayed, minimum 4 to 5 days will be required, but some immunity can be lifelong. You have taken measles vaccine, only one single dose, and that gives a lifelong immunity. Similarly, we get several infections and we develop lifelong immunity. I do not know where from I am coughing. This point is not written in your book. Interaction between PRR of the host and PAMP of the microbes because of the sharing of the antigen. You note down this point, I will explain what is this thing. Interaction between PRR of the host and PAMP of the microbes, but here interaction occurs between the antigen or haptens. It can be microbial, it can be non-microbial also, because you see we are allergic to many pollens, dust. These are not actually microbes, these are non-microbial. And the antigen, that is Ig, this is coughing, I am coughing. Suddenly I got exposed, maybe there is some allergen. While coming from the hostel till morning, before coming out from my room, there is no coughing. So I have been exposed to some kind of allergen. I need to take some drug, control it. So memory, there is no memory. Adaptive, yes, it is there. Cells involved, remember all these cells. Here natural killer cell NK cells, NK lymphocytes, monocytes, macrophages, dendritic cells, neutrophil, all of you know, but in addition all these cells, eosinophil, basophil, tissue mast cell, epithelial cells. In the thing, it is adaptive, that is B or lymphocyte or T cells. Other components are there, but you may not require, like complement, soluble, pattern recognition. You forget about this thing, but at least you must remember all these things. Now, all this I will explain. Have you completed? You just do a little fast, otherwise I will not be able to complete. There are many slides where and you have to write down many things. Some are there in the book which you are not required to note down, only listen. Be a little fast as if you are writing in the examination. And note down the points which you cannot understand. Ask questions, no issue. And you try to remember this thing. This was the question in the second prop. That's why I am written it in the beginning itself. At the end, you will not have any energy, you will not note down. Okay, just I am passing through, going through. So you just listen here, don't note down only this part. Basic purpose of immune system, sparing self and or eliminating foreign. Failure of doing this thing is causes diseases. Just note down that part. The basic purpose of immune system is sparing self, means I will not damage my own body. No organism, no animal wants that the body to be immunized will be destroyed by its own immune system. And we have to eliminate foreign materials. That is the basic purpose. And how do we do it? That is a very complicated mechanism. But if there is a failure that our body cannot recognize our own tissue, then we will develop diseases against our own body tissue. Might be our arthritis will be there, some joints will be there infected. My thyroid might be infected, my thyroid might be damaged, pancreas might get damaged, skin might get damaged, all autoimmune disorders that you will see in the clinics. So, design of the immune system, just note down these 5 points. One is specificity, there must be fine distinctions. See, if any molecule is there, suppose hapten molecule is there, small molecule, ortho, meta, para, benzene ring, remember. If an antibody develops against ortho position, and if you substitute that group into meta position or para position, it will not get. So, that is basically specificity. So, fine distinction is there. Ortho, meta, para, benzene. If you substitute the same group, the antibody developed against one ortho position will not react with the meta position of the same component. So, that is a very specific fine distinction. So, diversity. We are having a broad repertoire of antigen specific receptors. Means our body has evolved over crores of years of evolution, and during that period of time, we have encountered many antigens, and our body is having all the receptors which the body has encountered, and these are all present there in each lymphocytes. In many lymphocytes, and one lymphocytes will be very specific for particular type of antigen. So, this is an innumerable antigens, you can imagine. It is not a single day development, and the development is ongoing. Whatever immunity we are having, it will increase after 1000 years or 500 years. Some more things will be there, some things will be eliminated because maybe these are not required. Memory, as I told you, it accelerates the next immune response because we have developed an immunity. Vaccine, you have taken booster dose, initial dose and booster dose. What is the purpose of booster dose? To accelerate immunity. Demobilization, that means clearance. Once the fighting has over or the immune reaction is over and antigen has or foreign body bacteria or pathogenic bacteria has entered into the system, our body immune response was active, neutrophils were recruited, organisms were destroyed. Now there will be demobilization, clearance that so far no more bacteria neutrophils is required. What is demobilization? I am telling you, suppose there is a war, we are having an army, the army will be deployed into that place. Once the war is over, the army is no longer required. to be placed there. So, they are demobilized again to their own places. Or maybe we require excess amount of person to fight with our invaders. Once that crisis is over, so they will be cleared from that place. That is what is demobilization. And distinguish self from non-cells, I have been repeatedly telling, to prevent attack of normal cells of our own body. Now, I will give you 3 definitions of antigen. Remember whatever is suitable to you. First definition you can write down. One is an antigen is a substance which invokes immunological response. You note down any one you can write, but this is not a complete definition. An antigen, it is easy to understand. Only write down this part, an antigen is a substance which invokes an immunological response. The simplest definition. And causes the immune system to produce antibodies against it. But immune system antibody is not the only issue. Cell mediated immunity is there. So, therefore, antibody is not the only purpose. But just for understanding, priming you, specific, special, as I told you, ortho, meta, para. So, it's minute change in the protein molecule or any antigenic molecule will cause immunity, will act as an antigen. Suppose you might be allergic to milk protein, but all milk proteins may not be allergic. Maybe from something from dairy you are taking the milk and you are developing allergy. But if you take from the cow's milk directly from the the cowmate's house or from the cow itself, you may not develop the allergy. So, small differences in antigen structure is a recognition. Blood group antigens are there. Note down, A group of blood encodes a glycosyl transferase that produces A antigen, and N-galactosamine is the immunodominant sugar in the A blood group, which is present on the RBC membrane. B allele encodes a glycosyl that creates the B antigen, and that is D-galactose. Now, all of you know that if I give transfusion of A group blood into B group, there will be immunological reaction and there will be chance of death. Why? Because A group blood contains anti-B antibody, B group blood causes B antigen but anti-A antibody. Now, the moment you give B group blood into A group blood, there will be severe cross reaction, severe antigen antibody reaction. Because so that is the reason you can see why we don't give mismatched blood, but the same group blood. So it binds with the antibodies very specifically, and there is also a T cell receptor. So this is a better definition. You see, this one you try, if you can remember this thing, that will be very much useful. Antigens are foreign or self molecules. Even self molecules can act as an antigen when there will be failure, that are recognized by the adaptive and innate system. I will explain what is adaptive. I have told you the difference. That results in immune cell triggering, T cell activation, and or B cell antibody production. That covers almost everything of antigen. So that is a complete definition, and those who cannot remember anything, at least you tell a substance which induces detectable immune response, and within the bracket you write down humoral or cellular. At least no marks will be deducted, at least some concepts are there. But these will definitely attract the attention of the examiner. There is something more you know, and you know much better than somebody else or your counterparts. So antigen, anything cannot act as an antigen. There must be some characteristic features of an antigen. So note down just whatever I have written in the red, that you write down. Low molecular weight proteins are not, substances are not antigenic. Large molecular weights are highly antigenic. Protein and polysaccharides are good antigens. Structural diversity, protein with 20 amino acids. As I said that the low molecular weight proteins are not antigenic. We require some large molecules. So, polypeptides containing L amino acids are antigen. Foreignness, only antigen that are foreign and non-cell. Tissue specificity, antigen specificity is stereochemical, stereospecific. I have told you ortho meta para. Tissues of all individuals in a species contain tissue specific antigen. Suppose in your body, in our body, liver will express some antigen, kidney will have some express some antigen, or other organs are having some antigens. Gene is present in everything, DNA is present in everything, but all the DNA encoding the genes are not available in all the cells. RBC you will get hemoglobin, but hemoglobin is not present in other tissue. In the muscle you will get myoglobin, that myoglobin is not present in other tissue. So, that basically gene is expressed. So, each and every tissue is having some specific proteins. Forensic application, in recent tragical case, everybody know that we want a biological specimen available from the site of crime. So, blood antigen can be there, body fluids can be there, from cells can be there. Basically, we want to identify is the same criminal or not. The alleged person is there, whether it is matching with the alleged person or not. So, that is test is done by antigen, and in case of basically forensic nowadays DNA testing, we really go for DNA testing for matching, and HLA haplotyping is actually done. Classified antigens are blood group antigens, as you know. Histocompatibility antigen I will definitely tell, because these are required for your clinical purposes. Blood group antigens are required to know regarding transfusion. HLA antigens are required for organ transplantation. There must be matching. Haptens, so note down what is hapten. What is the difference between hapten and antigen? Haptens are not basically a large molecular weight protein, but they are small molecular weight. They are capable of forming an antibody and can become immunogenic with a large carrier. Maybe I told you that I have developed an immunoassay for progesterone. Progesterone was a very small molecule. It could not, if I inject progesterone into a rat, it will not develop antibody to what I needed. I coupled with bovine serum albumin, that is a very large molecule, 67,000 molecule with progesterone, and then I injected into the rabbit. So, the rabbit mounted immune response. From the rabbit, I collected the blood which contained antibody against progesterone. So, haptens you note down, a small molecular protein which by itself is not immunogenic, but can act as an antigen when it is coupled with a large carrier. So, suppose this is an antigen. It is a big structure. So, all this part of the antigen is not actually react with the antibody. A small part which reacts with the antibody is called epitope, and these are antigenic determinant. Means against this thing antibody will be produced. And suppose this is an antibody is always represented with an Y structure, and that reacts with the antibody. So, this is called epitope, and that part of the antibody which binds with that epitope is called a paratope. So, epitope and paratope basically are involved in antigen antibody reaction, not the whole compound. Antibody is again a large compound, large molecule. This is also a large molecule, but interaction takes place only between the two, just like a handshaking type of thing. The whole body does not get involved, only the hands are involved. It is just like that. So, epitope and paratope. Histocompatibility, just note down this part, very important thing. So, there are three classes, three types of antigens. One is class 1, class 2 and class 3. Remember this point, class 1 B, C and A, it is present in the 6th chromosome. Class 2 is basically class 1 and class 2 is important, I am telling you. I will explain. DR, DP and DQ, there are 3 locus and these are studied, actually studied before bone marrow transplantation, before organ transplantation, whether they are matching or not. The importance is that the class 1 antigen is present in all nucleated cell and platelets. Class 1 antigen, that means B, C and A. Class 2 antigen, DR, DP, DQ, these are present. Note down this, I will explain at one point of time. B lymphocyte, macrophage and activated T lymphocyte, how these are involved in the immune process, that you will understand. A function of the class 1 and class 2 molecules to bind and present to immune cells, this I will explain at the end. So, close linkage of HLA system, they are called, they are inherited en bloc. We inherit HLA antigens from our parents en bloc, means there are several alleles that are linked together and we inherit that. Whether he is my biological father or biological mother, or whether I am the actual biological child of that parents, that are decided by that HLA close link antigen, and that is what is involved in DNA paternity testing, disputed paternity. So, diagnostic importance of HLA, it is a MCQ question. HLA B27 is commonly done for ankylosing spondylitis, DR34 is for insulin dependent diabetes mellitus, allogenic transplantation. Suppose I am suffering from some bone marrow disease, bone marrow needs to be transplanted. So, who will be a donor? Either my parents or my siblings. So, we are basically transplanting from one human to another. That is called allogenic transplantation. So, siblings are better than parents. Why? Because parents can contribute only half of that component, but siblings can inherit both the things. So, all the siblings can be a better donor for organ transplantation as well as for bone marrow transplantation. But in our settings, we are having a 1 or 2 child. So, really we do not get any opportunity to get a good donor. So, we had to take some unrelated donors, as there is a high chance, as you can imagine, there will not be HLA matching and there will be tissue rejection. So, parents at the end, if there is no sibling, ideally mother or the father donates that bone marrow or kidney to their own child. So, DNA paternity testing, as I told you, because of that HLA link genes are inherited en bloc, using that thing he can be detected whether that is the biological father or biological mother, because that raises a legal issue during adoption cases. Even in your clinical practice, you will see that many persons are not having child, so they are childless couples, they adopt. Now, legal dispute comes at the end, and that is solved by this HLA testing, haplotype testing. So, vaccines, all of you have taken, only note down this thing. Vaccine is an immunobiological agent that produces antibody or protection against a given disease by producing antibody and other immune mechanisms. So, many vaccines you have taken, but the mechanism, the immune responses are not same. The difference between the vaccine and an infecting agent, that during vaccine preparation, the pathogenicity part is destroyed. We take the virus, only polio virus we eat, but we do not get infected. That is attenuated, or its virulence, infectivity has been decreased, so that it will no longer cause any disease, but it will mount an immune response. So, there is a live oral vaccines polio we are taking, but if you take that wild type, and it is the natural, what is basically available in the nature, which is not attenuated or which has not been destroyed, which pathogen part is not destroyed, we will suffer from polio. So, vaccine basic purpose is to retaining immunogenicity, but without any pathogenicity. We can have live vaccines, that means live virus we can take, we can kill that virus, and we can have a toxoids. You know that polio vaccination are given to the mass in general to create herd immunity, HERD, means a group of persons will be involved so that the entire population is immunized. So, there is no chance of the polio virus to be transmitted or available in the society. So, if the entire Bolpur persons or the entire West Bengal persons are immunized with polio, the polio virus is excreted through stool. So, all the persons who will be passing stool will have no polio virus. If by chance there is a single case of polio virus, it will cause a panic attack to the government. And I worked in polio lab for 6 months at Sanjay Gandhi Postgraduate Institute of Medical Sciences, and I knew what are the things that are done. All samples which were found to be polio positive, these stools were brought that are cultured, then we want to know what is the genetic structure. So, that is the basic purpose. The herd immunity confers to a mass population, and that is the reason why smallpox has been eradicated by mass vaccination. One day wreaked havoc. So, there are different types of vaccines. I am not going into the detail. So, these are common vaccines which you have taken as a healthcare professional. Hepatitis B will be given to you and you will be required to take, but I think all of you have taken. So, because of the active vaccination program, smallpox has been eradicated, but all the other diseases are not eradicated yet. Some infections are there. So, that is why we continue to give vaccination, but smallpox vaccination we do not give. So, there are various processes involved in vaccine preparation and there is desirable criteria how a vaccine is to be produced. This is a big chapter. Last year I took a full class on that. Remember one thing, Edward Jenner was the pioneer worker for vaccination, and the first vaccination of smallpox was done in 1796 on James Philip, who was 8 years old. It is a pioneering work. Even criticized his life. You can hear the life history. I can tell you one milkmaid suffered from some illnesses and Dr. Jenner doubted that he was having some smallpox. Then that milkmaid told Dr. Jenner that I cannot have that milk, smallpox, because I have already had chickenpox. So, chickenpox confers immunity against smallpox. That how milkmaid knew. And Dr. Jenner got the information from that lady, and finally he succeeded in developing a vaccine. So, many persons will tell many things to you. Please listen carefully. All are not saying rubbish things. Some good informations are available. So, that lady was so much adamant that I cannot have smallpox because I have contracted chickenpox. That means she had an observation that who suffered from chickenpox did not have smallpox. That observation was very much important. So, coming to this now class, basically with this prelude, it is already 40. Innate or natural immunity, this is available. I told you, respond immediately to foreign cells particles and acquired a specific adaptive immunity. As I told you, it requires time to develop. So, innate immune system, now write down what is PRR and PPV. So, this is innate immune systems. Basically, we are born with that innate immune system. What is that? It is an ancient immune recognition system. The host cells, note down, bearing germline encoded pattern recognition receptors that recognize pathogens and trigger a variety of mechanisms of pathogens of elements. Because we inherit the germline genes from our parents, and our parents had that genes. So basically pattern recognition receptors means our immune cells are endowed with that particular set of genes which is capable to identify that he is a foreign he is foreign, sorry, so these are the cells involved on pattern. I have told you in that differentiation, that table, the natural killer cell, monocytes, dendritic cell. Now there is one point, you say interaction between PRR and PAMP. You just open that that table. So PRR is present in human being. We are having that PRR in our immune system. The cells are having PRR, just like this one. Suppose this is PRR, pattern recognition receptor, or this is we are having this one, and PAMP. See, we are having this pattern recognition receptors. Oh, we are all having that thing. Similarly, microbes, pathological organisms, they are also having some antigen which is conserved, and they are present. That is called pathogen-associated molecular patterns. They express this thing on their surface. Suppose somebody is coming with arms, everything, or some animals are coming, you know that animal is dangerous or that snake is dangerous. So they are expressed, some snakes are coming with a hood, you know that is a poisonous snake. So that is what is PAMP. So that is present in the microbe. So the interaction takes place between the PRR and the PAMP. So immune system, this innate immune system doesn't require any exposure. They know by virtue of their property that this is a dangerous molecule. So this is pathogen associated, means pathogenic material are there, not non-pathogenic. Non-pathogenic do not express this thing. So that is a danger signal. So they are expressed by a group of microorganisms as recognized by the host PRRs. Host means we are host, and that is the offending agent in the mediation of immunity. So the moment our body encounters that PAMP structure within our system, our lymphoid system or the immune system is having sensors. They are scanning, they are moving in the entire body and simply moving, circulating where there are foreign particles. Just like our army or border security force, something like that, CISF, we are having several armed forces. So they are moving around, the police, they are policing the entire body. But the moment they get an offending agent, they simply arrest. That is what is that thing. So the innate immune system, what it can do, it can kill directly. Immediately it will attack, and it can destroy, encounter at the border level. Somebody entering into our system, into our country from other country. Just for example, I am telling you, and there is a standing instruction from the government that you simply encounter, kill it on the spot itself. Somebody is coming maybe with an identity of some sort of, intention to destroy our country. So that is called encountering, to kill directly. Or slow down the infection in concert with dendritic cells, I will tell you. That means you arrest that person. So that will be slowed down. So this is just like an RPF, innate immune system. It is very prompt and rapid. Something has happened at Bolpur, suppose railway station. So whatever police force is there in the local police station will simply move to that particular place, and they will call for an emergency to the district headquarters. So rapid action force will come into the action and control the situation. Innate immune system is just like that. I am just giving you an example so that you understand better. So that deploys a range of relatively non-specific yet effective way of containing infections. Suppose police is normally used for controlling any criminals. But civic police are there. Several other politicians are there. They are having some persons. You can contact that politician. There is a crisis. He will bring some persons. So this is not specific one. Immune system is innate immune system is like that. So acquired immune system, note down this part. It is an immune system mediated by T and B lymphocytes. And note down this first two parts sentence. Immune response by these cells are based on specific antigen recognition by clonotypic receptors. These are not available in your book, I am telling you. Clonotypic, what is clonotypic? That is the unique antigen receptor gene arrangement in nucleotide sequence corresponding to a particular identical position. It is very difficult to understand, but note down at this stage so that you can read it later, you understand better, or you ask when the class will be complete. When you read something, I will explain once again what is this thing. It is difficult, I appreciate at this stage, but you note down. So, what is clonotypic receptors? And this is expressed in all cells. There is one more cell that is called antigen presenting cells. These are involved in the adaptive immune system. So T cells, B cells and antigen presenting cells. And clonotypic receptors are involved in that. So in that chart you can see again, you look back, it is usually 4 to 5 days after the innate immune response. It is highly specific. In contrast to that non-specific one of the immune system, it is primarily mediated by the B and T lymphocytes and displays specific receptors on the plasma membranes. As I told you, we have been developed over crores of years of evolutions, and all the pathogens or all the antigens which we have encountered, we are having a receptor on our lymphocytes to all those things. The moment they get exposed to that particular antigen, that particular lymphocyte undergo clonal expansion. That means that particular cell will undergo expansion and it will generate immune response. So, it display recognition and binding of antigens, triggering and proliferation of different cells, and that great increase in the number of particular antigens, I will tell you, through a process of write down the clonal expansion. So that clonal expansion will basically result in high amount of antibody. From the picture I will show this thing, I will tell this thing, that will be easier. So after infection, first innate immune system is operative, and that is immediate, and then it will give the adaptive system to give some time. That 4 to 5 days time is there. So immediately during that 4 to 5 days, innate immune system will try to destroy or control that infection. Even it can exclude also. I mean, the entire system can be, the offending agents can be handled by the innate system if the infection is less severe. But if it is more, then innate system alone will not be able to do that thing. And that 4 to 5 day time, by that time adaptive immune system will come down. Example I told you, there is some problem at the Bolpur railway station. Police station came from Bolpur railway station immediately to control whatever it can do, and it called the district headquarters. So that time, that much time, so say maybe 2 hours will be, or 1 hour will be required to get some police from Siuri to Bolpur. So that is what is adaptive immune response. So they are interdependent, but they work in tandem. This is a district headquarter, even that is a state headquarter. If that is not possible, nearby districts, Birbhum adjacent district Bardhaman, they can call Paschim Bardhaman, they can call adjacent Murshidabad district also, send some force to Bolpur. So we are unable to control it. So they work in tandem. So as I told you, this part now, this I have shown it to you. You see that there is a physical barrier, innate immune system is there and adaptive immune system is there. So they interact, work in tandem. One can do that thing, but one, if it is not able to do that thing, that takes the help of other. And usually immune system operates in both the things are operating, that immune system and adaptive system, they are interdependent, then work in tandem. So types of immunity, one is humoral. Humoral means related to body fluids. Note down this part. What do you mean by humoral? Humoral means related to body fluid. It's a common term. As cellular means cell mediated. That is T cells mainly. Humoral is basically immunoglobulin, that is B cell mediated. Humoral is related to immunoglobulin, related to B cells, which are produced by the B cells. B cells get converted into plasma cells. From plasma cells, we get immunoglobulin. But cell-mediated immunity, T lymphocytes are improved. That is called T cells. And we can get a combination of the two also. Because as I told you, just like innate and adaptive immune system, work in tandem. Similarly, T and B cell also work in tandem or independently, as the case may be. Passive immunity means, it is active immunity means, suppose, just I am giving an example, you have been employed, you are working for the whole month, and at the end of the month you are getting a salary. Now that much salary is given by your parents directly at one time. That is passive one. You have not done any labor. So passively you got the money. But when you are in job, you have worked 30 days or say maybe 26 days in one full month, and at the end of the month you have earned that thing that you have acquired. Nobody has given it to you. Now passively you can get things. So normally we get during our intrauterine life from the mother. Mother gives that immune protection to the baby, and in your clinical practice you will give injections. Suppose you require to control the infection very fast. There is exposure to contaminated with the soil, with many materials, and you are suspecting that tetanus can occur. So in that case you will not only give injection against the bacteria, but you will also give immunoglobulin against tetanus. So specific normal human immunoglobulin can be given, specific human immunoglobulin, and in our days this normal immunoglobulin was not available. Anti-sera from horse, these were used to be given, and there used to be a reaction. So I could not come to that point. So immunity from the mother to the baby, this is most important. देखो तो ये देखो तो क्यों ऐसे किना पहले हमको बोलबो बंद कर दो एक बार जाओ तो देखो तो बाहर सारे ऐसे किना. So immunity from the mother, just note down this particular point. While in the womb, the passive immunity is transmitted from the mother to the fetus across the placenta. At a protective biological shield তৈরি করে the mother gives a protective biological shield, because baby is most important to the mother. So somehow it will try to leave no stone unturned to protect the baby. So high levels of immunoglobulin, IgM, especially IgG, of the cord blood and maternal plasma is transferred to the immune mothers. So a large proportion of the baby who are breastfed remain free from infection. So that is this I am stopping. Maybe I will not be able to cover in the earlier class, but you just read the immunology part from your book. In the next class, I will try to cover all these things, because I had to stop, I had to go slow. The subject is as such complicated. She has somehow told 3 classes. If required, I will take 3 classes also. Because I am not going to go fast, you will not understand. If I go very rapidly, honestly speaking, you will not understand anything. Even today, you have learned many new terminologies. To digest that thing, to internalize, it will take some amount of time. So, in your book, I am telling you all these things what I have told are not written. Whatever has been written in your book, you just remember this thing, and I will tell you something more. My basic purpose is not the first year. My basic purpose is your 5th year class, NEET examination, and to become a successful practitioner. And for that, I am priming you with the knowledge. This knowledge you will remember, I am telling you, while you will do the MD or PG, that these were taught in our class in the first year itself.